Hi, and thank you for attending this talk in the OHBM 2021 Open Science Room. I'm Amanda Mejia from Indiana University, and today I'm going to tell you about a tool that my group has developed to make it easier to work with CIFTI files. So the name of this tool is CIFTI-Tools, and it's an R package that allows you to read, analyze, visualize, and write CIFTI files all in one place. And I have to give major credit to my postbac researcher, Damon Pham, who is responsible for a lot of the content of CIFTI-Tools. So the goals of CIFTI-Tools are primarily to provide a single unified tool for reading, writing, visualizing, and analyzing CIFTI and its compatible GIFTI data in one place. And secondly, to facilitate the analysis of cortical surface as well as subcortical data using the advanced statistical techniques that are available within the R computing environment. So before I tell you about what CIFTI-Tools does, I want to tell you about a nomenclature I'll be using throughout this talk, and that is the CIFTI object class. So CIFTI with an X refers to the combination of essentially CIFTI data with GIFTI surface data. So we have cortex left metric data, cortex right metric data, and subcortical metric data, all of which is combined typically into the CIFTI format, as well as cortex left surface geometry and cortex right surface geometry, along with some metadata that's typically included in CIFTI and GIFTI surface files. So all of this together is known as the CIFTI object class within the CIFTI-Tools environment. So the first main function of CIFTI-Tools is reading and writing. So to read CIFTI data, we can use the read CIFTI function, aliased as read CIFTI, and we can pass in a CIFTI object as well as optionally GIFTI surfaces to create a CIFTI object within R. And then we can subsequently use the write CIFTI function to take that CIFTI object and write its contents to a CIFTI file, as well as to GIFTI surfaces if they are included in the CIFTI object. A second major function of CIFTI-Tools is visualization. So since CIFTI objects consist of both cortical and subcortical data, we have two different tools for visualization. The first is titled View CIFTI Surface, and it's a function that we pass in our CIFTI object to, and that comes up with an interactive surface-based visualization that can also be written out to a file for static images for publications, for example. In this example, we are using some surfaces that were read in along with the CIFTI file that correspond to maybe an inflated subject level surface. But if we don't provide surfaces, there are default very inflated surfaces that can be used for visualization instead. And later I'll show an example through a demo of these types of visualizations. For the subcortical data, we have View CIFTI Volume, which can create either a static or an interactive image showing the subcortical data overlaid on an M&I template or a provided structural image. Some additional features that are available within CIFTI-Tools include some of the processing steps available through the Connectome workbench, specifically resampling and smoothing. So if we take a CIFTI object, we can use smooth CIFTI to create a metric smooth version of that CIFTI. So here you see an example where you can see that the data has been smoothed a bit relative to the original image. We can also resample the CIFTI metric surfaces as well as the geometries, the surface geometries, to a lower or a higher resolution using resample CIFTI. And so you see here kind of an illustration that the data has been resampled to a lower resolution. And so we have fewer vertices indicated here on the surface. and so that surface geometry as well as the metric data have both been resampled. And we can also perform resampling of the subcortical data. Additionally, CIFTI-Tools provides a number of functions in order to do kind of manipulation and mathematical operations. So for example, we could add together two CIFTI objects to create a third CIFTI object that would contain the metric data would have been added to each other. And so here we have an illustration of that. We can also do univariate transformation. So for example, here I'm showing an illustration using absolute value. So this is known as an S3 class in R. We can basically apply any univariate transformation, such as absolute value or square root or log, etc., directly to a CIFTI object in order to perform that transformation just on the cortical and subcortical metric data. So here you can see that whereas previously we had some negative values shown in cool colors, here we only have positive values, all shown in warm colors. And then we have a number of additional manipulation functions that kind of mimic some of the things available in the Connectome workbench. So we have combine CIFTIs, which combines two CIFTIs that have different brain structures. For example, one may have cortex left, another may have cortex right, and so we can combine the two. We can also do merge CIFTI, so we can concatenate CIFTI column-wise or combine different columns from different CIFTIs. We can use new data CIFTI to replace the metric data of a CIFTI. Remove CIFTI removes a particular brain structure, such as the cortex right or the subcortical. And then finally, select CIFTI can be used to subset or reorder columns of the metric data in a CIFTI. So these are just a few examples of the math and manipulation tools available within CIFTI-Tools. Okay, so how would you actually go about using CIFTI-Tools? So first, it's important to download and have installed a copy of the Connectome workbench on your local computer or wherever you're working. And that's because we depend on the Connectome workbench for some of the background features that are key to things like writing and reading, as well as certain processing steps. And so once you have Connectome workbench installed, you should install CIFTI-Tools within R. So you can use install.packages to install from CRAN, or you can install from GitHub for the latest development version of the package. And then within R, this is the case for every package in R that's not loaded by default. You'll need to load the package each time you want to use it through the library function. And then each time you load the package, you'll need to set the path to your installation of the Connectome workbench. So you do that through the CIFTI-Tools.set option path like so. Okay, so now I'd like to show you a short demo, and this demo will focus on primarily reading and writing and visualization, but you can find an expanded version in vignette form at our GitHub page for this package, and that will include more explanation as well as more illustration of certain processing and manipulation functions as well. So with that, I'm going to switch over to my R Studio. Okay, here we are. All right, so I'm going to just step through this code. So I'm going to start by just setting my working directory, which is typically what you want to do at the start of any R session. And then here I have a line that will install the CIFTI-Tools package from GitHub if it's not already available on your system. I already have it installed, so that install is going to be skipped, but the library will be loaded that way. And then we can go ahead and set our path to the Connectome workbench. So for me, the workbench is located inside of my applications directory. And all I have to do is point to the workbench, which is like the super directory of the Connectome workbench once you have it installed. Okay, so we're going to be using some sample data that's available through the CIFTI-Tools package. So we can access that data in this way. And I'm going to show you the names of those files. So here you can see that we have 4 files, including a DT series, a dscaler, a dlabel, file, as well as another descaler file. So these first three only contain cortical data, and this final one contains cortical and subcortical data, and it's just all ones. Also notice that these first two have the same kind of prefix to the name. They actually contain the same data, but they have a different intent, so descaler versus DT series, and also the descaler has been resampled down to a resolution of 6,000 vertices per hemisphere, and that is simply because of limitations on R package size. So these two files have been resampled down to 6,000 vertices per hemisphere from the kind of standard 32,000 vertices per hemisphere. So we'll be playing around with this data through the demo. I'm going to also point to a couple of GIFTI surface geometries. These are group level, group average inflated surface geometries at 32,000 resolution. And so first I'm going to illustrate the reading and writing features within CIFTI-tools. So we'll be first playing around with a descaler, which is this myelin and cortical thickness 6,000 resolution file. And so I'm just going to use read CIFTI and not specify any additional arguments. And then I'm going to show you that CIFTI object, just a summary of that CIFTI object. So you can see here that it's indicating that the CIFTI object contains the left cortex and the right cortex metric data. And then within each of those, it tells you how many surface vertices and how many columns the left and right surfaces contain. And here there's been no GIFTI surfaces provided. We'll see later how the summary would illustrate that GIFTI surfaces have been provided, surface geometries that is. And then you can also see here that the intent is printed as well. So now I'm going to read in another CIFTI file, but I'm going to include the left and right surface geometries. So I'm going to be now reading in the DT series file, which is this full resolution myelin and cortical thickness file in DT series format. And then I will just run read CIFTI by providing that CIFTI file name as well as the two GIFTI surface file names. And now when I print out a summary of that CIFTI object, you can see that it contains left cortex, right cortex brain structures as before, but now it also contains left surface, right surface brain structures. The left cortex, again, you can see the number of vertices as well as the number of columns. And here it states the left surface model is present. And that's because we passed in that GIFTI surface file corresponding to the left hemisphere. And then similarly for the right cortex. And now the intent is listed as a DT series. Okay, so that's an example of how we can read in GIFTI surface geometries along with the CIFTI file, but we can also add them in later. So here I'm going to first read in the CIFTI file, the DT series, using read CIFTI, and then I'm going to use the add surf function. And I'm calling this new CIFTI object XII2. You can see that it seems to have very similar information. The summary is equivalent, and I can use the all.equal function to show that those two ways of adding in the GIFTI surfaces are indeed equivalent. So I can also write out CIFTI and GIFTI data to files. So to illustrate this, I'm going to first just get the list of all the files in my current working directory. And then I'm going to use the write CIFTI function on my CIFTI object, which again includes the surface geometries. So I'm going to specify the name of my CIFTI file to write out the metric data, as well as the name I want for my GIFTI surface geometries. So I'll call this write CIFTI function, and you can see it prints out some updates. And creating that combined CIFTI file from the components is the slowest step there. And now I'm going to show a list of the new files that have been created in my working directory. So you can see here, these are the files that have now been written to my working directory through this write CIFTI step. Okay, so that concludes the portion of the demo on reading and writing. And so now we're going to turn to visualization. So first, the CIFTI-tools package does depend on a few additional packages, but you'll be prompted to install those as needed in certain cases where you may not need them in all for everything you may want to do. So an example of that is the RGL package. This is not installed by default when you install CIFTI-Tools. So if it's not installed, this line will install it for you. Otherwise, this line will load the package. So for surface visualization, again, we have the view CIFTI surface function. And I'm going to illustrate here a few of the primary arguments, including color mode, index, zlim, widget, and fname. So I can simply pass in my CIFTI object to view CIFTI surface for a default visualization. And so you can see a pop-up window will show up. Actually, I don't think you can see that, so I'm going to share my entire screen. Okay, so this pop-up window now shows up, and hopefully you can see that it can be rotated. It has this default color scale. The palette here is a sequential palette because we only have positive values in our data set. And now I can, I'm going to show you how I could change a couple of the settings. So I'm going to change the limit of the color scale to using the zlim argument. And I'm also going to specify that I want a diverging palette. And I'm going to give the plot a title. Okay, so now you can see that I have a diverging palette that has cool colors on the bottom half of the palette and warm colors on the top half of the palette. This will be used by default whenever we have negative and positive values in our data. But we can also specify it if this is the palette that we want to use, even if we don't have negative values in the data. And again, this is rotatable. Notice that these surfaces correspond to the inflated surfaces that were provided when this CIFTI object was read in. And so I could also have used the default surfaces available if my CIFTI object did not contain surface geometries. So now I'm going to read in a CIFTI and not provide any GIFTI surface geometries. I'm calling that object XII no surf. And so if I call view CIFTI surface on XII no surf, you'll see that we have some very inflated surfaces that are included in the package and are used by default whenever the surfaces are not specified by the user. And here I'm also going to illustrate how I can specify and kind of slide through multiple columns of the data. So here we'll see a couple of pop-up windows just momentarily pop up and then close out. And then a widget's going to show up in this plot window. So here you can see we have a widget with a slider. And I can slide through to go through the different columns that have been provided. You'll see that the title changes as well as the surface data that's been plotted. Alternatively, if I wanted to plot a single column or multi-columns, but I wanted to write that to a PNG, I could specify the fname argument, and that's going to write out each of the columns that I specify as a PNG file. So again, I'm going to record the names of the files that are written in my working directory, and then I'm going to call this function with the fname argument provided, which will write out one file for each column that I've specified, and those windows will close out. And so if I call this line, it's going to tell me what new files exist in my working directory. And you can see that there's two files that were created, starting with the my IMG name that I provided, but then ending with the actual name of the column. Okay, so I've shown you so far D-scalar and DT series visualizations. I'm also going to show you a D-label, which is going to use a qualitative palette by default, and we'll come up with a legend in this window. So we're going to have our normal pop-up window showing the qualitative D-scalar surface visualization. And then you can see here in the background in the plot window, we have a qualitative legend that we can later save if we wish. Okay, so that was a brief overview of our surface visualization tools. Now I'm going to show you a couple of volumetric visualizations using this dscalar1s file. And so I'm going to use the view CIFTI volume function. And you'll see that by default comes up with an image of 9 different slices. I can set the number of slices. I can also set the zlim and the number of indices to control this a little bit more of these details. And then also I'd like to show you just finally that we can also set up an interactive graphic, basically building on top of Papaya and the Papaya R package. And so here we have a widget that we can kind of zoom through to the different slices. And you'll see that in the console, this prevents our R Studio session from being able to kind of continue as normal. So we have to stop that widget before we continue with our analysis. But we can also kind of get around that by opening up a new window where we see we have that interactive graphic, and then going back to R and closing that out. And then our other window will still persist and be able to be visualized. Okay, so that basically concludes the content of the talk. I just wanted to mention a couple of other R packages that we've developed that are compatible with CIFTI files via dependent on the CIFTITools package. The first is BayesFMRI, which can allow you to do GLM analysis for task fMRI. It has a spatial Bayesian model implemented, but it also has the classical massive univariate traditional GLM approach implemented. Template ICA-R can allow you to do group ICA, dual regression, as well as template ICA, which is sort of an alternative to dual regression that uses empirical population priors for better estimation. And then finally, the fMRI scrub package, which performs scrubbing via a measure that we've proposed called leverage, as well as DVARs and traditional head motion measures. So you can find all of those tools available at my GitHub repository. And so just to conclude, a couple of final thoughts. If you do use CIFTITools, we would greatly appreciate it if you would acknowledge that in your publications. And you can find a citation using R's built-in citation function and just pass in the name of the CIFTITools package. The package may need to be loaded for that to work. And then just finally, if you have ideas for additional functionality, this package is it's not in beta mode, but it's definitely continues to be under development as we add new features. And so if you have ideas for functionality, please reach out to us. If you run into any issues or have any questions, please also feel free to reach out. And you can reach both me and Damon either through the GitHub page for CIFTITools or just directly by email. And so with that, I'd like to thank you so much for coming to this talk and for your attention and your interest in this work. I'd also like to thank my funding agency, NIBIB, and you can reach me a number of ways, GitHub, website, or Twitter. And thank you again.
Hello, my name is Eric Werner, and I am with the Center for Translational Research in Neuroimaging and Data Science in Atlanta, Georgia. Today I will be speaking about COINSTAC vaults for federated machine learning. The open science movement seeks to make scientific research accessible to all levels of inquiring society. This includes open access, open data, and open to society. By promoting these goals, open science can bridge the STEM gaps both between and within countries and accelerate science to meet global challenges. However, as with many endeavors, there are conflicting goals. We want to make data open, but also preserve the fundamental right to privacy for participants in scientific studies. In the United States, there are 18 identifiers that are covered by the Health Insurance Portability and Accountability Act, or HIPAA, known as Protected Health Information, or PHI. Neuroimaging datasets often contain some of this information, such as addresses and scan dates, and must be anonymized before being made public. However, the matching of anonymous data to publicly available data to identify an individual, known as re-identification, can compromise the privacy of research participants. Because of the risks of re-identification, many datasets are never made public. In 2019, researchers at the Mayo Clinic did a study on re-identification of participants using head MRIs and photographs. They collected both structural MRI scans and 5 photographs from 84 participants. Then they reconstructed the face of the participant from each MRI scan using an automated system and used publicly available facial recognition software to match each reconstructed photo to a real photo. The facial recognition software found the correct photo for 70 of the 84 participants, or 83% of the time. The authors of the study did not use techniques to deface the image, noting that such techniques are often not used in anonymized datasets because they reduce the utility of the images. This was a powerful study that shows the danger of re-identification in neuroimaging. If people believe that their private information is not safe when they participate in a study, it will be harder to recruit participants, and data quality will suffer. Participants will be less likely to report information that can possibly be used against them. Breaking the trust between scientists and participants harms science. The Collaborative Informatics and Neuroimaging Toolkit for Anonymous Computation, known as COINSTAC, seeks to strike a balance between open science and privacy by enabling open, reproducible data analysis while guaranteeing privacy. COINSTAC allows researchers to perform federated machine learning and coordinated pre-processing using containerized software. COINSTAC is open source software available on GitHub under an MIT license. It is a cross-platform application built with Electron, which can run on Mac, Linux, and Windows. It has a user-friendly interface that allows users to create new consortia, set up and run pipelines, and view results. The COINSTAC application is built entirely in open-source programming languages, using modern JavaScript frameworks, such as React and Apollo for the front end, and Node, Express, and MongoDB for the back end. COINSTAC utilizes the MQTT communications protocol, which was invented for the Internet of Things, to guarantee reliability in uncertain network conditions. Computations are run inside of Docker containers and encapsulate all dependencies, including operating system, software libraries, and custom code. Any programming language can run inside, including Python, MATLAB, and R. COINSTAC enables federated machine learning, which allows us to use data without directly accessing it. Imagine a situation where there are multiple sites that have data that they cannot share. They also want to analyze their data together to build more accurate and generalizable models. COINSTAC allows them to build a model together without sharing data. There are multiple ways to do this, including single-shot and multi-shot algorithms. With a single-shot algorithm, each site builds a model on its own data and then shares the model with the remote node. The remote node then combines those models into a global model and shares it back to each site. In a multi-shot algorithm, a model is built iteratively using an optimization routine like stochastic gradient descent. On every step, the remote node shares a global weight vector, and each local node returns the gradient on its dataset until the algorithm converges. Differential privacy is a technique that can be used to more strongly prevent re-identification. The idea is that noise is added to the results of an aggregated query to prevent a malicious user from identifying individuals by making multiple queries and cross-referencing with public data. With differential privacy, the probability of re-identification can be quantified and dialed up or down, depending on the importance of performance versus privacy. The data owner can keep track of how much data has leaked from a site, known as its privacy budget. Differential privacy can be combined with federated machine learning. Two ways to do this are output perturbation and objective perturbation. In output perturbation, noise is added to the output of an optimization algorithm, whereas in objective perturbation, noise is added at every step of the optimization algorithm. Objective perturbation generally works better but requires good local feature mapping and identical pre-processing across sites. We have implemented differentially private support vector machines and logistic regression. Each site trains its own classifier, represented as the W vector in the figure, and sends it to a private aggregator. That site then combines the outputs of the classifiers to build a final classifier that performs better than each individual model by itself. As you can see, the combined classifier achieves results that are significantly better than what can be achieved at a single site. This differentially private model achieved a classification error rate of 5%, whereas individual sites achieved error rates of over 20% on neuroimaging data. Many other studies have shown similar results. For example, this paper presented a federated neural network classifier to differentiate smokers from non-smokers in real fMRI data. The federated model achieved results that were close to results achieved using pooled data and were far better than results from isolated sites. You can see in the figure that the pooled model attained a classification accuracy of about 80% after 130 iterations, whereas the federated model achieved accuracy of roughly 74%. Isolated sites performed the worst, with accuracies between 35 and 50%. Similarly, in a paper recently placed on bioRxiv, brain age estimation was performed using support vector regression on 3 types of data: structural parcellations from FreeSurfer, gray matter volumes, and functional network connectivity matrices. Brain age estimation algorithms are first trained to predict the actual age of the subjects using neuroimaging data, and then applied to estimate the age of new subjects. Large gaps between estimated brain age and actual age have been shown to be biomarkers of Alzheimer's disease. Our federated method achieved results that were statistically equivalent to centralized models in all 3 cases. Because neural networks often have millions of parameters and require many iterations to converge to a solution, there is an important need to minimize the information they share with other sites in a federated algorithm. Recently, we have published new methods to minimize bandwidth, including efficient distributed autodifferentiation, or EDAD. These methods decompose gradients into lower rank matrices before transmitting them back to an aggregator node, drastically reducing communication bandwidth. EDAD reduces bandwidth by multiple orders of magnitude, as seen in the figure on the left, with no sacrifice to accuracy, as seen on the figure on the right. This is an important optimization that will speed up the training of neural networks on neuroimaging data. We currently have many federated algorithms that researchers can use to study both structural and functional imaging modalities. We have differentially private methods and are working on secure multi-party computation algorithms, which do not require data to be decrypted throughout the algorithm. In addition, we are working to add differential privacy to our existing federated algorithms. We are also in the process of adding linear mixed effect models, imaging genetics models, multimodal models, and neural networks with optimizations to reduce communication overhead. These computations all implement algorithms that have been or will be validated and published in scientific journals. Additionally, they are all open source and available on GitHub and Docker Hub. Now I would like to show you some example results from COINSTAC. Here we have a result using federated regression on structural MRI images. The images were pre-processed using voxel-based morphometry, and then a regression model was created for each voxel with covariates of age, gender, and diagnosis. The output is a map of the regression coefficients and their significance for each covariate. COINSTAC also features a dynamic functional network connectivity pipeline. Functional MRI images are pre-processed and then used for federated group independent component analysis and dynamic functional network connectivity. After group ICA, federated Mankova and univariate statistics can be run as well. We also have federated methods to visualize high-dimensional data. A federated version of T-stochastic neighbor embedding can be used to quickly identify data quality issues and see patterns in the data. Here you can see the results of a toy example using the MNIST handwritten digit dataset. Each group consists of images of handwritten digits. The algorithm uses a nonlinear mapping to project each image with many pixels onto a two-dimensional plane, while preserving distances in the original high-dimensional data. We are actively working with multiple groups. We have published a study with GIGA and are currently in the process of publishing another. Going forward, we are investigating the effects of both genetics and environment on the brain, with them using COINSTAC. We are also working with the ENIGMA schizophrenia and bipolar working groups, looking for transdiagnostic biomarkers. With Penn State, we are developing a federated algorithm for site harmonization, called COMBAT-DC. Finally, the Child Mind Institute is working with us to develop COINSTAC vaults, which are the subject of this talk. Earlier this year, we presented a paper with GIGA on structural changes in brain gray matter linked to age, smoking, and BMI in adolescence using datasets from Europe and India. This analysis used over 2,000 structural MRI images collected at 14 different sites across 2 cohorts. The images were pre-processed using VBM and then analyzed with a federated regression algorithm. Results showed a widespread reduction in cortical gray matter and increase in the hippocampus with age, bilateral increases in the hypothalamus and basal ganglia with BMI, and cingulate and thalamic decreases with smoking. This is consistent with and extends previous findings. We did another study with GIGA in which functional MRI data was used to examine differences in independent component spectra and functional network connectivity associated with alcohol and tobacco use. The study used over 2,000 subjects in sites in India and the US. We ran federated group ICA with the NeuroMark template, followed by federated ICA statistics on the component spectra and functional network connectivity. One of the benefits of the spatially constrained ICA approach is that we gain the advantages of estimating subject-specific, maximally independent spatial maps and time courses. But the constraints facilitate automatic component selection and ordering. This then enables us to fully automate the spatial ICA approach. The results of this analysis reveal that both tobacco and alcohol both increased high-frequency power in the spectra, in the blue. Alcohol also shows reduced power in the 0.05 to 0.1 Hz bold frequency range. FNC results show mostly reductions for both alcohol and tobacco, as seen in the figure on the right. We are currently writing the paper and plan to submit it for publication this year. You can learn more about the NeuroMark template by reading the paper by Du et al. At the bottom of this slide. We are working with ENIGMA on multiple fronts. We are using COINSTAC to study the relationship between positive and negative symptom scale assessments and features in structural, functional, and diffusion brain images. We will also run multivariate approaches, such as group-independent component analysis and source-based morphometry to help understand schizophrenia. We will also use multimodal machine learning approaches to predict symptom scores on the basis of multiple neuroimaging modalities. Additionally, we will apply the same techniques to data from bipolar subjects, as they often do the same assessments as schizophrenia subjects, and search for commonalities between the disorders. ENIGMA has had great success doing meta-analyses manually thus far, and COINSTAC will allow them to run a greater range of machine learning techniques on their data. After running studies with multiple groups, we have noticed several challenges related to federated learning. The research consortia that we have worked with have sites around the world, in North America, Europe, and Asia. Because COINSTAC operates synchronously, and all sites need to be online simultaneously, it is often difficult to find a time that works for everyone. It is also hard for some people to leave their laptops on and connected to the internet for some longer pipelines. To solve these issues, we are creating COINSTAC vaults. These are persistent COINSTAC instances set up at a site or in the cloud, which are always automatically available for federated computations. To bypass manual involvement, each vault has a list of approved computations and covariates in its dataset that it allows for group analysis. Instead of coordinating runs ahead of time, other researchers can simply run COINSTAC and add the vaults to their consortia. Additionally, we will put in place consortia of vaults organized by various themes, such as the type of disorder they have data on, or the type of analyses they are willing to run. For example, we can have consortia organized around schizophrenia or Alzheimer's disease. Similarly, we can have a deep learning consortium, where each site has good hardware resources with GPUs that can be used for training neural networks. COINSTAC vaults will have many benefits that support the goals of open science. They will reduce the time and effort that researchers spend orchestrating federated computations. They will also be a part of open source scientific infrastructure and enable rapid reuse of data and reproducible research. In this video, you can see how COINSTAC vaults work. First, the user creates a consortium as usual. Next, he is given the choice to add a pipeline to run. Notice the number of choices he has for computations. He is also able to select a cloud user, which is a COINSTAC vault, to join the consortium. After selecting the vault, there is only one pipeline that he can choose, which has been allowed by that node's owner. Additionally, only covariates allowed by the owner of the vault can be added to the computation. This gives the data owner fine-grained control over his or her data. We are working with the ChildMind Institute in New York to develop this technology and host their data on COINSTAC vaults. We are also establishing a procedure by which more sites can set up, configure, and register their own vaults for federated learning. We will post updates on our website and social media platforms when we are ready to accept more sites. We have many plans for the future of COINSTAC. Once we are ready to scale, we want to grow our network of COINSTAC vaults and consortia. We also want to improve interoperability with other federated machine learning platforms, such as PyGrid, and add more methods using differential privacy and secure multi-party computation. In the near future, we want to strive towards more fairness. And by that, I mean follow the fair data principles by making results findable, accessible, interoperable, and reusable. We intend to do this by annotating COINSTAC results with rich metadata and giving them DOIs. We also want to give sites a way to post metadata and a way for users to search for other sites on the basis of that metadata. Finally, we want to move towards using bids for both inputs and derivatives. We are always looking for more collaborators. Our codebase is open source and on GitHub, and we could use more software developers to contribute to the application. Likewise, if you are a neuroimaging researcher who works in a consortium where data sharing is an obstacle, please look into COINSTAC. We may be able to help you. Finally, we are constantly adding new federated algorithms and pre-processing methods to COINSTAC, so we welcome algorithm developers to add new algorithms to our platform. If you are interested in collaborating or just want to learn more, you can visit our website at coinstac.org, view our GitHub repository at trendcenter/coinstac, or email me at ewerner@gsu.edu. In closing, we are grateful to be supported by the NIH on these grants. Thank you for listening.